Now, before we start talking about the next class of macromolecules, which are known as lipids, I want to address the concept of hydrophobicity because it'll be central to understand the biological properties of lipids. Lipids are a very special class of molecules, and they're special in many ways. One of them, and one important one that has a huge impact on the cell and the possibility of having cells, is the fact that lipids are hydrophobe. Now, I want to explain what hydrophobicity is and what it is not. One thing, let's look at this here, the word hydrophobicity. So hydro, of course, pertains to water, and phobicity, well, pertains to a word that we associate with, phobia. And we know phobia is, we know that it's like a fear of something. So if you are someone that is not comfortable in large crowds, you might suffer from agoraphobia. If you are uncomfortable in small spaces, you might suffer from claustrophobia. So we associate phobia with a fear, a feeling, a very unpleasant feeling. Now, when we're talking about molecules, well, molecules do not, of course, they don't have feelings, but they have behaviors, and that's what we're going to look at right now. So when we're talking about hydrophobia in the case of lipids, we're not talking about someone that's afraid of water. I'm talking about a molecule that is a hydrophobic molecule, which means that it will not interact with water. If it's hydrophilic, then it will, because this is the opposite of phobia. So a hydrophobic molecule will have a tendency to stay away from water, and it will interact with other hydrophobic molecules in order to stay away from that water. Okay? So this is what I want to explain here. Now, before I get into this, I want to bring back the notion of this very special small molecule that is known as water. So when we look at water, we know that there is an oxygen, and we know that there are two hydrogen atoms. And the other thing that we know is that oxygen has a tendency to pull on the electrons. So basically, there is a little charge here, not a real charge per se. It's just that there's a portion of this molecule, water molecule, that has a little bit more of a negative environment to it, because electrons have a tendency to spend more time here than they will there, right? So those deltas here, remember, those are partial charges. They're not real charges. It's just that they have a tendency to be more positive on the hydrogens and a bit more negative where the oxygen is, only because of the way this molecule is structured and the way hydrogen and oxygen interact with one another. These are still covalent bonds, right? But it's just the way the hydrogen, the electrons will spend their time in this tiny molecule here. Now, we know, because we've seen it, that ions, so for example, sodium, which is a positive, will have an attraction for this, this oxygen here. And we know that the ion chloride will be more attracted to this environment right here, right? So we know that. So we know in this case that whenever there is a charge these charges have a tendency to interact with water. They want to interact with water. So in this case here, this is a hydrophilic environment, right? This here, sodium chloride, is of course hydrophilic. It will interact with water because of the way it is configured at the electronic level. Now, when we're looking at lipids, lipids will have this structure that are long chains of carbon. So what we have is carbon, 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 etc. And we know that these carbons have the possibility to form 4 bonds. And in the case of lipids, these bonds will be linked to hydrogen atoms. Okay? 4 bonds, right? So let's count that. We have our carbon here, and we have bond number 1, bond number 2, bond number 3, and bond number 4. And if we look at this one, we have 1, 2, 3, and 4, etc., etc. Okay? What's interesting here is that the electrons in every single one of those atoms is distributed equally. It's part of the covalent bonds that we find everywhere, right? So these covalent bonds are not non-polar covalent bonds. Remember, if you don't remember that, please go back into this lecture where we were talking about polar covalent bonds. These here are non-polar covalent bonds. So basically, the electrons are distributed equally, so nobody here, or none of these bits and pieces here, have any partial charges. There is no delta negative or delta positive. None of that exists right here. Okay? So, what happens? If we mix this with water that has a charge, a partial charge on every single one of the atoms, well, there is no attraction between the two. Because water wants to, because of the charges, it wants to interact with positive or negatively charged molecules, but in this case, there's no such thing. So lipids, which are formed of long carbon chains, will not interact with water. Water will not interact with those two, with these lipids here, these long chains of carbons. That causes a hydrophobic environment. Now, don't get me wrong. It's not that water doesn't want to interact with this molecule here. It's not that it doesn't want to. It doesn't want anything, right? Molecules are molecules. They don't decide anything. But they don't see the lipids. They just don't see it. There's no way they can detect it because there's no charges to interact with. So we know that there is no connections between the two here. So now what's going to happen when we have an environment that has both of those together? They won't mix. So think, for example, if you're making a salad dressing, you have vinegar that's mostly water, and then you might add olive oil on top of that. And as you mix them, you see that there's a separation between the two of them. So if you have a beaker or a glass or a bottle, so what's going to happen is that you're going to have something like this. So this is my beaker right here, and at the bottom I might have my vinegar, which basically is water, and on top of it I will have my oil, and we know that the oil is going to separate and float on top of the water. Now, the important part here is not that it floats on top or under. It doesn't matter where it floats. What matters, though, is that there is definitely no interaction between the two of them. They will be on top of each other or below, right? And they are not interacting. Now, if you have only a few drops of oil in a glass of water, so this is our same beaker here. Let's look at my beaker right here. Then if I'm looking at this beaker from the top here, so this is my top, this is what I see right here, right? What I have is if I have water at the bottom and I add only a few drops of oil on top of that water, we know that this oil is going to form these droplets right there. And you might shake this all you want, the oil droplets will always form after. Why? Because all of the lipids that you disperse by shaking will eventually reconnect with one another, and water will float around that, will be around this, right? Pushing basically the lipids together again. And they always form because they don't mix, and they don't mix because of the hydrophobicity of that. So now, from now on, when we look at lipids, we need to keep in mind that lipids are mostly hydrophobic. Okay, we'll see that we can introduce a few changes in those lipids to make them a bit special, with maybe a small piece of it that's hydrophilic, but lipids will remain hydrophobic for the main part. So that's one thing that you need to really understand, is what hydrophobicity is in order to understand the properties of these very important molecules that are lipids.